All right, here we go. Welcome to Stat's virtual event, Cancer's Breakthrough Engine, sponsored by Scorpion Therapeutics. Thanks for everyone joining us. This is actually Stat's last event for the year. So we're excited to end on this high note. I'm Adam Feuerstein, senior writer for biotech at Stat. I'm sure you all know how to do these virtual events right now. At the bottom of your screen, just a couple of housekeeping notes. At the bottom of your screen, you will see a chat function. You can use the chat function if you have any issues with the technical issues, if you need to talk to the staff at Stat, or you can also chat amongst yourselves. More importantly, we want you to use that Q&A function. If you see the Q&A tab there, the button, use that to ask questions of our guests today. I will try to incorporate as many questions as I can into our conversation. So type your questions. I will see them on my screen and I will use them. With that, before we get started, I want to welcome Axel Hoos, I'm sorry, Chief Executive Officer of Scorpion Therapeutics, for a few sponsored introductory remarks. Axel, welcome. Oop. Axel, I believe Axel, I think you're muted. Oop, Axel, I think you're still muted.
How is this?
Oh, there you go. All right. I'm going to do that
All right.
again. Axel, how are you?
All right. Well, thank you for the kind introduction and the
Go ahead.
opportunity to participate in this event and actually sponsor it. So, we're all here because we share the same ambition of making transformational progress for cancer patients and give patients the opportunity to, certainly impact massively on their disease or eventually cure it. And I know the C word has been difficult to achieve for many diseases over the last several decades. I'm seeing progress now that I would term precision oncology 2.0, or the next generation of cancer medicines that are using genetic anomalies to treat cancer very effectively. And we have seen great progress since we began this process. And, the two obvious examples to point out are Herceptin, the first approved antibody for precision oncology, and then Gleevec, the first approved small molecule for precision oncology, both dating back anywhere between 20 and 25 years. And we have seen many generations of improvements in that area, particularly through the introduction of new modalities. So, for example, go from antibodies going to antibody-drug conjugates, or bispecific antibodies, or effective combinations of small molecules and antibodies. And so have seen real transformational benefit. In the small molecule space, for Gleevec, we have seen 6 generations of small molecules targeting BCR-ABL to make incremental benefit, having led us up to now about a 10% cure rate of this disease that was previously fairly fastly progressing before we had access to these small molecules, and has then, led to the detriment of the patient. So we made progress here, dramatic progress, and I think we're at the verge now of seeing the next generation of medicines come, particularly antibody-drug conjugates and new small molecules designed through a new era of integration of different scientific approaches and technologies that make the molecules better. So recent examples, certainly are the ALK inhibitors in lung cancer, we're all familiar with. The first generation of ALK inhibitors, crizotinib, eliminated chemotherapy from the treatment of those patients in the ALK positive population. And then now, in the next generation, with drugs like lorlatinib, we could improve over the first generation with hazards ratios of as good as 0.28. So clearly, massive steps in the right direction by integration of new science and technology that focuses on higher selectivity, new modalities, and then eventually deeper insights in the target biology that gives us, access to the right targets at the right time. So I think the next generation that we are now going to see will be, PI3K inhibitors. There's several of those on the market. Initially, they were developed on the basis of either just targeting the pathway or then subsequently going after isoforms that have a more differentiated expression in different cancer types. But we haven't gotten yet to really a mutation-selective targeting approach that might enhance the selectivity, and with that, allow us to open the therapeutic window and target these diseases that express those mutations more effectively. So I see the next wave of, precision oncology to begin with mutation-selective small molecules, and then, of course, continue with the next generation of ADCs, like Enhertu, for example, with the great effects we have seen in breast cancer. So I look forward to, an insightful discussion here about what will the future of precision oncology hold, and how we can all, work together to optimize the outcomes for the patients we want to serve. So I think I'll leave it at that with my introduction. I'm honored to be a sponsor of this panel and look forward to the conversation.
Axel, thanks so much for that introduction. And without further ado, I would like to introduce our rock star panelists today. Speaking today, we have Dr. Vivek Subbiah of the Sarah Cannon Research Institute. We've got Dr. Suresh Ramalingam from the Winship Cancer Institute of Emory University, and Dr. Patricia LaRusso of Yale School of Medicine. She's also the president-elect of AACR. Vivek, Suresh, Patricia, hello. How are you?
Very well, very good. Thank you.
So Pat, I want to start with you. When we were doing our prep, you told me that you're old enough to have trained both Vivek and Suresh, which of course means that you are wise and you have tons of experience. That doesn't mean that you're old. So maybe set the table for us a little bit here and tell us kind of like how far you feel like precision medicine in cancer has come, since the approval of Gleevec. Axel had mentioned Gleevec 20 years ago. How far have we come?
Yeah, so, I started working before we had cloned the human genome and been able to use the genome to help us in terms of identification of potential targets for the treatment of cancer. Actually, when I started, we didn't have cell phones, and we had much cursory computers than we have now, like the one I'm using to talk to everyone today. I think that the cloning of the human genome set the stage to really transform one of the avenues in oncology, if not multiple avenues in oncology, in terms of diagnosis, treatment, prevention. I was an attending when they were developing Gleevec to not only treat CML, and it transformed, as Axel said, a disease that killed people in a few years to allowing them to live a sustainably long life and be able to utilize second and third generation agents. And then GIST, which my mother died of within a few weeks of diagnosis, which Gleevec and subsequent generations of that agent or that target have really transformed those patients with that disease. So yeah, I can tell you that precision medicine, it's come a long way, and it's probably today what I consider more exciting than ever before. And one of the reasons that I stay doing what I do, the times are more exciting today than they've ever been.
And Vivek, let me turn to you. What's on the top of your to-do list of when we think about precision cancer medicines? What's still left to do?
Oh, we have a lot of work to do. I think we are just starting our work on precision medicine. Again, as Pat said, 30 years ago, they said we cannot sequence the human genome. Then we sequenced the genome, which cost like $3 billion. And now we can get it as down to $1,000 in the US. And colleagues in India and China can get it done for $100. one colleague in Singapore and another colleague in Mumbai have challenged that they're going to take it down to $1 in the next decade. So genomics as diagnosis with $1 is going to be done universally, right? So the last 30 years, we have witnessed breathtaking, unparalleled advances in scientific research, from better understanding the pathophysiology of disease, unraveling the cellular processes at atomic resolution. And, at the same time, we have had exponential, growth and explosion in developing therapies that alter the course and outcome of diseases, not just in oncology, in all areas of medicine. We have seen improvements in genomics, immunology, better understanding of proteomics, metabolomics, gut microbiomes. We are just still scratching the surface of microbiomes, epigenetics, oncolytic viruses. In parallel, we are seeing AI, artificial intelligence, big data science, computational biology, right? Also, recently, we saw the dawn of CRISPR-Cas9 technology that is opening up a tantalizing array of opportunities in precision medicine. So I think that this is just the beginning of what we are going to be starting to see as precision medicine. In the last decade, it's been a breathtaking tipping point. So we are in a tipping point here, but I think we are just still scratching the surface here. Again, so much to do in the next decade.
Suresh, when I think about tyrosine kinase inhibitors in particular, I think of like when we think about the sort of the evolve, how that's evolved. I think of Tagrisso, for instance, and I'm going to use the brand name because that's kind of what we do. Third generation EGFR inhibitor for non-small lung cancer, which tells you sort of how far we've come. You were involved in some of the clinical trials for that drug, and I wondered if we could use that as an example to kind of talk about some of the lessons that we've learned, in the development of these precision medicines, using kind of Tagrisso as that example.
Absolutely, Adam. I think it provides a great education in how we can be more precise in targeting specific driver mutations. Now, incidentally, next year will be the 20th year since the EGFR mutations were described in the literature for the very first time. And 2003 was, if I recall right, the first year where a TKI was approved for lung cancer. That was gefitinib, and then erlotinib followed suit. These were not selective. These were reversible inhibitors. And when we started using them, we were giving it to all patients with lung cancer without knowledge of what drives the subset of patients who derive the most robust benefits. Now, after the mutations were described, it still took us about 5 to 7 years before clinical trials would show that this group of drugs only works for patients with mutation and should not be given to patients without mutations. While these drugs benefited, we saw very quickly that, similar to what happens with Gleevec and CML, the gatekeeper mutation, in this case it's the T790M mutation, drives resistance in a matter of months in nearly 60% of the patients. And that really called for development of agents that specifically target T790, but also the original exon 19 and 21, which are the two most common activating mutations. But we didn't get to from first to third directly. We did study a group of irreversible inhibitors that were not mutation specific. And we said, if reversible is good, irreversible inhibition of the target should be even better. So we flooded the receptor with these drugs. They work, but they also have high levels of toxicity because they don't distinguish between healthy tissue and tumor tissue. And that's where the third generation drugs made such an important difference, because you have a high degree of selectivity to the mutated receptor. So the toxicity profile is much better. We first showed that you give this group of drugs in patients after they have developed acquired resistance to T790, to first and second generation, when they have the T790, and then it worked. Then the question was, if you knew the T790 was the main pathway by which these tumors develop resistance, can you give this drug up front and then shut down that pathway from becoming the escape mode? And that's what studies showed. So now these drugs are used in the first-line therapy for patients with metastatic EGFR-mutated lung cancer. These are highly selective, so toxicity is not an issue for a vast majority of the patients. Quality of life is great. And these patients go on these drugs for a year and a half too. These drugs also get into the brain very well. So you're also getting the benefit of activity against brain metastases, which is a common event in EGFR and ALK mutated lung cancers. So really, this shows the paradigm of drug development now has come to the point where we don't even try non-selective drugs anymore for new targets. You go directly to look at mutant selectivity. Can you get away with the mutant selective drugs? And that does not mean we've answered the problem. These drugs work, but eventually resistance develops. We're learning about resistance mechanisms to osimertinib. And now there are a number of novel approaches that are being pursued to overcome that acquired resistance. So we're still making progress, but we've come a long distance.
Pat or Vivek, maybe talk a little bit about kind of, next, when we talk about next generation, and as Suresh mentioned, particularly with resistant mutations and what we see, maybe talk a little bit, because that's a little bit more forward thinking if we think about it. Where's the field going?
So, three things we've learned. Again, since we started from Gleevec, let me talk about the Gleevec. Timing is vital in cancer therapy, right? How was CML successful? Because CML has three well-defined stages. One is the chronic phase. Another one is we call the accelerated phase, and the blast crisis. So the major steps that enabled this transformation was discovering of the underlying genetic defect, which is the BCR-ABL gene, identification of a best-in-class targeted agent, imatinib, that obviated the enzymatic activity of the BCR-ABL, and administration of this drug in patients with newly diagnosed disease. That has transformed. So the third step, treating patients with early disease, is one that is most not frequently addressed in solid tumors. So for instance, in CML, in the chronic phase, we are able to cure patients, and as Pat said, we are patients live for a long time, and again they live their natural they die of natural causes, not because of their CML. But even today in 2023, if a patient comes in with blast crisis, and if we administer imatinib, the responses are around 15 to 20%. I think that's what we are seeing in solid tumors, although it's not well defined in solid tumors. I think that's what we are seeing in solid tumors, that we are treating later and later stages of patients with these precision drugs. And it may be a paradox that these drugs work even in the later line of therapy. So the timing is vital in cancer therapy. I think if we strike earlier in the disease course, I think we may have a better success with these precision drugs. Another thing is resistance. Again, resistance develops. I think cancer follows the story of the Hercules and nine-headed Hydra, right? So Hercules tried to cut off one head of the Hydra, and two heads pop up. So that's what we are facing with tyrosine kinase inhibitors in oncology. So we go after one target, and we have two resistance mechanisms popping up, and so on and so forth. So cancer at the molecular level is a malignant snowflake. And the next decade, what do we need to do is treat patients earlier in the disease, come up with best-in-class agents to go after that specific target, work specifically on on-target resistance mechanisms, and also, again, one wild card is the off-target resistance mechanism. If you see most of the tyrosine kinases, each patient's resistance mechanism is unique. So how are we going as a field to come up with a program to treat every one of those patients with customized combination therapies? I think that's where we have a lot of work to do.
Pat, do you have any thoughts on that? I mean, particularly, on the resistance side, and it seems like we're sort of chasing these resistant mutations, right? We can't predict which mutation is going to impact a patient beforehand, right? We know that after the fact.
Yeah, so a couple of things to follow on with that, as well as what Vivek had discussed. So I think he's right on with solid tumors in hopefully treating them earlier and earlier in their disease course. So we've seen that over and over again, the differential in efficacy between a drug for many of these solid tumors, utilizing a drug in the advanced metastatic setting versus the adjuvant setting and the neoadjuvant setting. And I think more and more we're seeing those trials of neoadjuvant treatment. But talking about personalization of disease. In fact, I was talking to one of the patients in clinic yesterday. I don't know two patients that have ever really had the same tumor. Even though we know a lot about the genomics of the disease, what we don't understand yet and are starting to learn, are developing the necessary tools to help us, is the functionality of the genomics. I don't know that any genomics in a patient, no two tumors are created equal. And I think it's important combination strategies, especially with small molecules, are going to be very important. But the old adage of how we develop combination therapies based on, got us, in early phase trials, if these two drugs in this dose are safe, you advance the other dose, then you advance, et cetera, et cetera. That is as unpersonalized as we get. I think the future is probably going to be in hopefully understanding not only the functionality of those alterations, those molecular alterations that we see, but hopefully in tumors, understanding the ratios of functionality to be able to better strategize the doses within the combinations that are going to help within each individualized patient. Problem again is overcoming toxicity. And that's where I think these next generation of agents are going to have to focus and be developed more on the genomics and obviously minimizing the toxicity, which we're starting to see that. We're starting to see that with PI3 kinase inhibitors and other targeted drugs. But the toxicity profile is changing with next generation drugs, lending themselves more to targeting the tumor less at a greater degree than targeting normal tissue.
I know we see, the approval of drugs, particularly in lung cancer, we're seeing the approval of drugs for rarer and rarer mutations. And obviously those all require that the patient's tumors be, we need to sequence them. We need to figure out the mutations that are driving those cancers. And I wonder from your perspective, and anyone here can chime in, is how accessible is that now these days in our healthcare system?
Well, it depends on whether you belong to the have or the have-nots. The reality is, Adam, even in the United States, not every patient with stage 4 lung cancer undergoes molecular testing. And we know there are disparities based on rural versus urban, racial background, education, literacy level. All of these matter. And there was a report just 2 years ago at ASCO where nearly 30 to 40% of the patients don't get the complete next generation sequencing. So we need to do a better job on making sure every patient is tested. The second part is access to these medications. This is not a given. Just because you found a mutation doesn't mean the patient is going to get the drug. There are a lot of barriers. Insurance barriers are key. And, I live in Georgia, which is one of the states that have not expanded Medicaid. So we see a lot of patients who have these mutations that do a lot of things to make these medicines doable. Some patients take it every other day to extend the prescription, right? Some patients take half the dose. So we have to do a better job of making sure that patients have access to these medications. Now, that requires changes at many levels. It's not just going to happen by any one of us wishing that this should happen, but it's going to be a concerted effort across the board.
Vivek, you had
even with patients that have decent health insurance, Adam, if a patient has a molecular alteration and there is a drug available targeting that alteration, but it isn't FDA approved for that particular disease, that patient may never be able to see that drug because their third-party payers won't necessarily approve it. And many of these drugs, for the vast majority of people, even in the United States, it's not affordable out of pocket.
And absolutely, I think, if this is a state of affairs in the US, when I give talks and meet with people and get emails from many countries, again, most of them don't have access to many of these precision medicines, right? So although patients should be the real epicenter and ultimate beneficiary of precision medicine, the complexity, cost, the Byzantine regulatory policies, especially insurance, prior authorization, competing priorities, lack of infrastructure, and access, right? Limit only a few major centers and few major geographies and few zip codes accessing these precision medicines. We talk in oncology about going to the moon, and I'm a clinical trialist, drug developer. We have access to all these, drugs in niche boutique centers, but majority of the world doesn't have access to these, precision medicine drugs.
Well, one of the other things, Adam, before you ask us the next question is,
Sure.
even if a patient has a molecular alteration, if it's not been studied in that disease before, and the diseases become smaller and smaller in terms of subpopulations, as we learn more and more about the molecular profile of the tumors. But if it's not been studied in that disease before, we don't know ultimately whether the patient is going to benefit or it's going to be a detriment to that patient. So what I tell my fellows and junior faculty, all that is, as an example, all that is BRAF is not created equal. That, BRAF has been around a long time. BRAF inhibitors in combination with MEK inhibitors or alone have been demonstrated to be active in melanoma. It's not that easy in BRAF mutant colorectal cancer. And we see that paradigm with different alterations across the spectrum of different diseases. These drugs don't work the same, even though a tumor may have a similar alteration as a different histologic subtype.
Is that a challenge for, we talk a lot about tumor agnostic drug, approvals across different tumors. Is that a challenge? I don't know, Vivek or Pat, is that a challenge to that kind of future?
So, absolutely. I think, there is a challenge and an opportunity. So tumor agnostic drug development, right? The first approval was, in fact, an immunotherapy for pembrolizumab for a genomic biomarker, MSI high microsite satellite instability high deficient mismatch repair cancers. Again, that opened up the doors for tissue agnostic drug development. And again, in my personal opinion, I would say every target is a pan target. Again, I think BRAF colorectal put a wrench for some time in the tissue agnostic drug development. But again, we needed a human's clinical trials to show that, right? So again, preclinical models at that time showed that BRAF V6, tumors with melanoma and colorectal cancers did the same with BRAF inhibitors. Only the clinical trials showed a striking difference of 60 to 70% in melanoma versus 0% in colorectal. At the same time, we went back to the lab and we found that there was an operating mechanism of resistance only operating in colorectal cancer. So we could add an additional drug to go after that particular resistance mechanism only in colorectal cancer to overcome that specific mechanism of resistance. So then, again, let's talk about BRAF, tumor agnostic. So unfortunately, what happened was because of colorectal, the field as such, right, did not develop that as a tissue agnostic drug. So they started developing that in lung cancer. We did it in anaplastic thyroid cancer. So we did a basket study called the ROAD basket study, the Rare Oncology Agnostic Research, where we enrolled, several different histologies, and we found that BRAF was actionable in many tumor types. So recently, last year, with a pooled analysis of ROAD, the NCI match study, and also another pediatric study, we were able to secure a complete, an accelerated approval in a tissue agnostic fashion for BRAF and MEK inhibitors. But let me tell you this point. So again, melanoma, the BRAF and MEK inhibitor combination, the dabrafenib and trametinib, was approved in 2013 and 2012, I believe. And in tumor agnostic fashion, it was approved in June 2022. So it took almost a decade to get a second indication, a tumor agnostic indication in many cancers. So again, this biomarker was approved in melanoma, but whereas a patient with any other cancer did not have access in the real world. So how many patients in the real world do have access to clinical trials? Think about that patient with salivary gland cancer in New Mexico who did not have access to a BRAF inhibitor. in 2015. Think about that glioblastoma patient in North Dakota who did not have access to those treatments. So again, what happened was colorectal cancer, drug development put a wrench in tumor agnostic. We've learned from that. So again, learning from that, what I would say is every target should be considered as a pan target. In addition to, once we pass the phase 1 of drug development, once we get a recommended phase 2 dose, in addition to pursuing in specific tumors, we need to have a biomarker-driven basket cohort. A human screening study will be the only answer to come up with a go, no-go decision if a drug works in that particular disease or not. I don't think so preclinical models will be ready to answer that question for that particular target. So again, my personal pitch to all whoever is listening is, in addition to tumor-specific, we need to have a tumor-agnostic cohort so that we can screen, we can have human screening studies. Otherwise, for instance, ALK fusion, again, they are tumor-agnostic targets. We don't have a tumor-agnostic drug. So in the real world, patients don't have access to an ALK inhibitor, right? They have to appeal insurances, and it's so hard in the real world to get access to these precision medicine. So again, that's my personal pitch for all those who are listening, is to add a tumor-agnostic drug, do human screening studies, learn, go back to the bench, and see if there are specific operating mechanism of resistance, and add a particular drug for that particular disease alone.
So let me, we have a question from the audience, Nir, and thanks for your question, Nir. And the question is, what combinations being explored are you most excited or optimistic about? You guys are all involved in, Pat and Vivek, you're certainly involved with early drug development. You do a lot of early trials. But yeah, what's out there? I know, Vivek, you were just at ASH, but maybe what combinations are out there that you think have a lot of promise? Anybody?
Yeah.
Wow, that is a $1 million question.
Yeah.
Maybe Ramesh and Vivek can help me. It's not about just exciting combinations. It's about specific combinations related to specific tumors. And we see some combinations where we are trying to overcome mechanisms of resistance in terms of upregulation of other targets, like Vivek was talking about, BRAF, EGFR. God, I mean, there's a lot of combination strategies. The challenge is to overcome toxicity. And some of the ways we're circumventing that right now is with bispecifics, as an example.
Let me ask you, maybe ask you a related question, because there's another.
Or IO. There's a lot of combinations.
There's a related question about whether or not we're ever going to get away from non-targeted therapies like chemotherapy. Now, I know we've seen combinations of TKIs and chemotherapy. I mean, what's the sort of consensus view, if there is one, of the panelists about chemotherapy?
So, let me answer that, previous question. So again, right now in oncology, one drug matched to a driver alteration may not be realistically expected to cure patients or achieve resistance in each and every tumor that has distinct and complex alterations. Other drugs are definitely needed to overcome the resistance. So again, it may not be a one-size-fits-all approach in oncology. Again, the question previously was about specific combinations, but rather the paradigm of individual therapy means that traditional way that drug regimens become standard of care no longer works, right? So if you think about, there are 300 drugs in oncology. There are approximately 45,000 true drug regimens and 4.5 million 3-drug regimens. The traditional clinical trial model design breaks down. So, however, I think, this conundrum is solvable because, 10 years ago, when I saw a patient with, say, 100 mutations, right? I was very sad for them. 200 mutations, I was sad for them. If I saw a patient with BRAF or ALK fusions, I would be happy because we get one target, one drug. But when I saw a patient with, say, 100, 200, 300 mutations, I said, what, how can I combine 100 drugs together? But today in clinic, if I see those patients with 1,000 mutations, I'm so excited because they are the best candidates for immunotherapy to work. Mm-hmm. So science moves on. We learn a lot about, some things which we will not be privy to, right? So again, the eyes cannot see what the brain does not know. And So so we may not be seeing. So with AI technology, right, we may be coming up with key hubs to target specific vulnerabilities in cancer that we may not be privy to with the previous technology. So again, hopefully we can go after those key hubs with specific targets so that we can overcome these challenges.
Suresh, I saw you shaking your head before.
Yeah, I was thinking about the point you made, question you raised, Adam, which is, can we get rid of non-targeted drugs like chemotherapy? And the answer to that is it doesn't look remotely likely that we are going to. And I don't even think that our objective should be to get rid of them. Our objective is to keep the patient alive for as long as possible and see if we can get a shot at cure. What we have learned from targeted therapies in the solid tumors is, as was mentioned, we're not curing these patients. So what is the best way to combine, sequence, alternate that will help us overcome resistance, manage persistence? We haven't talked about persistence as a problem because a lot of patients, even who have 95% tumor shrinkage, have the small population of persistent cells that eventually repopulate the tumor. And there are studies undergoing to understand what are the molecular pathways that drive persistence and see if you can target them even at the outset. An example, we just heard the results of the FLORA-2 study that was published in the New England Journal, where combining just age-old platinum-based chemotherapy with EGFR TKI results in improved progression-free survival by almost 10 months. Now, we don't know if the survival is going to be better or not. It's still not mature at this point. But this just shows that there are different population of cells that can be hit by varying strategies that we use. So I think we have to think about what's the best way to extend the ability. We've talked about, if you can't cure cancer, can you make it a chronic disease that patients can live with? And that is going to require all of the above approach, not leave one group of drugs behind.
And Pat, you were also shaking your head there.
Yeah, you've got to keep in mind, Adam, that with many of the antibody drug conjugates that are either commercially available or are undergoing clinical investigation and potentially soon to be FDA approved, many of them have as a payload a chemotherapy, a cytotoxic drug. So we're finding different ways to use cytotoxic therapy and to deliver it. And many of the antibody drug conjugates with the cytotoxic payload, I look at as just a different way to give that cytotoxic therapy, more like low-dose continuous infusion administration, especially with the topo-1 inhibitors like deruxtecan or exatecan. So, I think that as Ramesh said, cytotoxic therapy is going to be here, I think, at least during the time that I continue to treat patients. How it's going to be used in combination with other drugs, I think, is what we continue to explore and look for.
So, Pat, you mentioned antibody drug conjugates. I wanted to shift the conversation in that direction. Obviously, we've had the approval of the emergence of Enhertu, a new ADC, and ADCs, as you guys know, has been around for decades, and and it's another example of a class of medicines that we've improved upon and seen refinements and have gotten shown greater benefit for patients. We have another question from the audience along those lines, and Michael wants to know, data at San Antonio showed that sequencing of ADCs wasn't very effective, and so he wants to know: do we need better targets? Do we need better antibodies, better payloads? What does the future of ADCs look like?
Yeah, so I wasn't at San Antonio, but in listening to what some of my colleagues said, much of the sequencing was done, or the sequencing that was presented was in, you have a drug, you have an antibody drug conjugate, and you give it to the patient, and then they progress, and then you go on another antibody drug conjugate. But keep in mind that I believe, I may be mistaken, and Vivek, if you were there, please correct me, but I believe that the sequencing was done with different targeted antibody drug conjugates, but they had a very similar payload. So I think what we're now seeing is if you give at least in the arena of breast cancer, as was presented in San Antonio, if you give a particular tumor two different antibody drug conjugates, one for which they progress, and then a second with the same or similar payload, the lack of response is probably because they've already progressed on that payload, and that payload was a cytotoxic drug like a topo-1 targeted drug that, maxed out its effect as low-dose continuous infusion with that frontline ADC, and now you're giving the same payload in a second-line setting. Almost like giving cisplatin as an example, or carboplatin, and the patient progresses and you give them cis or carboplatin again. Even though you're directing a different target, the payload is the same. I mean, Vivek, Ramesh, maybe you can comment on that.
Absolutely, 100% agree, because, again, antibodies are like chemotherapy, right? So again, there was a recent paper published in Annals of Oncology, I believe, with Nectin-4 antibody drug conjugate with a TROP2 antibody drug conjugate. It was interesting because they had different antibodies and different payloads. And again, although this was an early phase study, this showed deep responses in patients with bladder cancer. Again, I think the next couple of years, we're going to be seeing more and more of those studies right at the get-go, not at progression, right together, dual ADCs sequentially at the same time, like I would say concurrent ADCs, dual ADCs, not at progression, not when the tumor develops resistance. So again, it's like combining two chemos with two different targeted agents. I think we are going to be seeing more and more of those drug designs, at least in the early phase, in the next 2 years.
But you're not only talking about different targets there, you're also talking about different payloads.
Exactly. You're talking about different payloads because it's giving two different chemotherapies, right? So, again, think about oncology as a field, right? Adult and pediatric oncology. What have we cured? Germ cell tumors. You have pediatric ALL. Pediatric ALL is we've cured pediatric ALL. That's standard of care treatment. Hodgkin's lymphoma, ABVD. So these are all combinations of different modalities of, chemotherapy, different types of chemotherapy, different mechanisms of action of chemotherapy. And I think with the ADC space maturing, we'll be seeing more and more of these ADC combination, ADC plus IO combination, ADC plus TKI combination. The key for us is how we can administer all these drugs and balance toxicity and also efficacy at the same time for these patients. Again, that's a challenge we have for the next decade.
Suresh, any thoughts on the future of ADCs?
Yeah, I think we're quite early in the ADC process, even though, Adam, you mentioned we've been around here for a while, because when you go back 20 years when these ADCs were developed, we didn't have any hits. We were just drawing a blank after another. And after a while, the enthusiasm just went away. Now that we have some drugs that are really good and hit specific targets, and HER2 being one of them, not just in breast cancer, but even in HER2 mutated lung cancers, we are now at a place where we know these drugs can be effective if you use it right, if you have the right payload, if you have the right linker technology, and if you identify the right patient population where these drugs are likely to work. So I think all of these questions that Vivek and Pat mentioned are very germane. They're all open questions. We need to understand why some patients benefit. For example, when you look at TROP2 targeted antibody drug conjugates, you see responses in 20-30%, but it does not connect with TROP2 expression. So then what is driving that 20 to 30% response? So we have a lot to learn. The good news is we at least know this technology, this group of drugs can be helpful for patients, and this is going to increase investments, which is going to ultimately lead to the next breakthroughs.
Well, I wanted to ask you guys all about quote unquote undruggable targets. And we've recently obviously had some success with KRAS G12C, which was once thought to be undruggable. We now have two KRAS G12C drugs approved, but I would, my perception is that maybe the efficacy of those drugs has kind of not lived up to the expectations that we thought they would be when we first saw the clinical data. And I wonder if you guys had any perspective on that and kind of both with that mutation specifically, like what's needed to sort of make, help that benefit more patients, but other sort of undruggable, quote unquote, targets.
So do you want me to answer?
Go ahead.
Can I start? Yeah. So talking about KRAS G12C, I know that, we're disappointed with the overall results of a drug like sotorasib or adagrasib because, we were thinking once we could drug this, we might be able to cure the patients. But first, I got to say that I'm actually excited that what we saw with those drugs, that yes, we were able to drug the target. And we did see some responses. But that was our first generation of drugs targeting G12C. I think we learned a lot. We're going into the next generation of drugs targeting G12C. I think what we're going to see is more exciting data on the next generation of drugs that are better, more effective, possibly less toxic in combination. And we're going to start seeing more combinations because one of the things about G12C, and Ramesh sees a lot of these patients with lung cancer, most of these patients just don't have G12C mutations or alterations. That's one of several other targets. And so we recognize now we can drug G12C. We're getting better at drugging G12C, and we're starting to really develop some of the combinations that may be able to overcome some of the resistance for more prolonged efficacy, at least right now, with hopes that we may be able to cure these patients, especially if we start treating earlier and earlier on. But I think that was the seed that was planted that showed we could grow a plant, but now we have to fertilize it and we have to make it stronger. Ramesh, you see these patients. What do you think?
Pat, you're exactly right. The modest efficacy that we're seeing with sotorasib and adagrasib doesn't mean these are not good drugs. It's the target as well playing an equally important role. When you have an ALK positive patient and you give them a TKI, the more recent generation of TKIs, the median PFS, we're now talking 3 to 4 years. Whereas in EGFR, with very similar drugs, the median PFS is 20 months. So the target to a great extent determines how effective these drugs are going to be because the biology of each target and how it influences cell signaling varies. I think we have scratched the surface of KRAS G12C. Now you're beginning to see multiple ways to approach KRAS G12C, and we saw some promising data with a drug that targets G12D, the RAS on, RAS off inhibitors. So the field is now taking off, and we're going to see better ways. However, we're going to have to study more about the impact of co-mutations. KRAS patients have P53 mutations. They have LKB1 mutations and KEAP1 mutations and many more. So how each one of these interplays with the KRAS mutation is going to determine the effectiveness of these drugs. And we're going to have to treat larger groups of patients to be able to slice and dice these things. But I'm not for one bit less enthusiastic about KRAS G12C inhibitors because the PFS results are not as great as what we would see in some of the other targets.
Yeah, no, 100% agree with what Dr. Ramalingam and Dr. Pat LaRussa have said. So again, in 2023, there are not undruggable targets, right? So again, superior chemistry and highly differentiated compounds are bringing us to go after all these targets. And again, KRAS G12C, if you see that New England Journal of Medicine paper about resistance mechanism, every patient has a different resistance mechanism. So how are we as a field going to provide these personalized medicine options for our patients sitting in front of us? Right? I think even though we get these alterations, still, even with the best-in-class ALK fusion and ALK inhibitors, we are not curing all patients. We are not getting complete responses in all patients. Again, as we are still performing a shallow personalized medicine for the generic patient sitting right in front of us. So how can we offer deep personalized medicine to patients? I think that's going to be the challenge in the next decade.
Mm-hmm. There's a question from the audience, and it's sort of related, but Jordan wants to know, what is the lower limit in terms of mutation, target prevalence, patient subpopulation size that spurs drug development? Are there ways to incentivize the pursuit of smaller and smaller populations?
I think we need Vivek and Ramesh to help with this one, but
yes,
one of the things that's been really, really challenging recently is the IRA that came about with the federal government to try to decrease the cost of drugs. And so with many of these novel drugs, it costs millions and millions of dollars for these companies to develop these drugs, from discovery in the lab to development in the clinic to FDA approval. And if you have a small subset, if you have a drug and you've first identified it in a small subset of patients as being active, with IRA, companies may actually now hold off on running to FDA approval because of the limited number of years now over and above limitations that were previous for patent expiration that allows them to recoup the dollars that it took to develop that drug. We think that companies are making lots and lots of money, but there's a lot of money that goes into developing these drugs. So we have to sometimes watch what we ask for, because if they can hold off by a couple of years to submit their NDA of the same drug, but has a larger patient population that will allow them to recoup the dollars that they've spent developing this drug, they may hold off. And that isn't good for patients, even though the cost of drug for the patient may ultimately be less. It's going to hurt, I believe, drug development. I don't know, Vivek, Ramesh, how you feel about this.
Yeah, I mean,
I'm concerned that small studies are going to be penalized in terms of getting those drugs out there first.
Yeah, I wonder if you're seeing the impact of the IRA and kind of from your perspective.
See, that's been the challenge, right? So again, two things. One, in the last decade, what has helped precision medicine drugs is the accelerated approval program, right? So accelerated approval program, as you know, was developed during the HIV pandemic, but more than 80% of the precision medicine drugs have been benefited by this accelerated approval program. So again, the accelerated approval program has benefited lung cancer. Again, Dr. Suresh knows this well. EGFR, ALK, MET, RET, KRAS, and all these drugs have been mainly, received accelerated approvals. So these have definitely corresponded to the sharp decline in population-level mortality in non-small cell lung cancer. Compare that to small cell lung cancer, where we have relatively stable population-level mortality. So non-small cell lung cancer has changed. So there was renewed hope for patients with rare diseases like FGFR2 mutated cholangiocarcinoma, RET mutated medullary thyroid cancer, RET fusion positive thyroid cancers, RET fusion positive many cancers. So early approval was also an important factor in terms of health equity, because all patients may not have access to clinical trial or expanded access protocols. So in addition, the accelerated approval also promoted the uptake of real-world biomarker testing, which was otherwise limited because of an absence of an FDA-approved therapy. So the accelerated approval program, provided early therapeutic access for many, many patients with rare diseases with cancer. Unfortunately, recently, what we are seeing early phase drug development is, not many companies are wanting to go for accelerated approvals for these rare biomarker-driven cancers like cholangiocarcinoma, like sarcoma, etc. They want a big indication. They want a randomized study indication. They want an earlier indication. So again, these are probably the unintended consequences of the law. Again, at one point, we want to make medicine cheaper. At the Other side, we want to make sure that these medicines are accessible to patients, right? So in genomic medicine, we have a law called Moore's law. The cost of genomics comes down from $3 billion to now $1,000. But unfortunately, in drug development, we have the opposite of Moore's law, which we call it Arun's law, right? So cost of medicines in 1960s, 70s was like 30 drugs. It took $1 billion. Today, in 2023, it costs $1 billion to make one drug, right? So again, these are the challenges which one person or we cannot fix. But unfortunately, because of unintended consequences of IRA, I think the rare cancer patients are going to be, it's going to be challenging for patient access to rare diseases in the real world. Again, hopefully, in the near future, this can be amended just for patients with rare diseases and for patient access.
Suresh,
with accelerated approval. That's the important.
We're getting right up on time. But Suresh, I wanted to give you the last word.
I want to say we have to be.
Ramesh, sorry, Suresh.
No worries. As the cost of developing drugs increases, we can't lose sight of the fact that we're not investing in a smart manner when it comes to developing drugs. How many times have we seen phase 3 trials initiated based on 15 patients worth phase 1 data? Pat is just doing the phase 1. The company has 5 phase 3 trials lined up before the results are ready. How many Me Too drugs do we have for every target, right? So I think we need to be smart about investing and follow the science. We should let science lead the business, not business lead the science. And if we do that, I think we can bring down the cost of drug development.
I mean, even in lung cancer, Adam, there's been a neoadjuvant trial recently that led to FDA approval of a drug. They didn't look at the biomarkers and listen to what the biomarkers had to say. So it's approved generically, but not every patient. There were subsets looking at the forest plots for which it made a lot more sense than others. So smarter studies.
And I, again, completely agree with what Dr. Ramalingam said, because oncology drug development has become like a fashion industry, right? So many Me Too drugs. If there's one drug approved in that space, there are plenty of Me Too drugs coming in that same space. I think would be good, smart investments, biology driving, science, biology driving drug development, also customized combination therapies, driving drug development. I think we all need to work together. I think we work in silos, but we all need to work together as a multi-stakeholder group, NCI, academia, industry, community, everyone, for us to get these precision medicines to the right patient at the right time. Thank you so much.
Yeah. So we are out of time. This has been a fantastic discussion. Vivek, Suresh, Pat, thank you so much for sharing your time and your insights today for our audience. I want to thank Scorpion Therapeutics for sponsoring this event. And of course, I want to thank the audience for showing up during the busy holiday season here. As I said, this is our last STAT event for the year, but of course, we are already planning STAT events for next year, including our first STAT Summit, which will be held in New York City on March 21st. And at that event, we are going to be gathering industry leaders and patients and discussing and trying to find real answers on how to make sure that science is benefiting people who need it the most. You can get more information about all our events on the STAT website. And with that, I will say happy holidays to all of you. And I hope you guys have a great break. And thanks again for participating.